Okay, I think we'll go on and get started. So thank you all for being here. This is great. I'm Edith Zemanek. I'm from the University of Colorado, and we're going to be talking about treatment of pulmonary infections today. So as far as disclosures, both Daria and I do not have any relationships to disclose for this. And so just to talk about our objectives, so we're going to talk about new approaches for antibiotic-resistant bacteria. We're going to talk about some results from phage therapies and discuss new treatment options for Pseudomonas and Burkholderia. And we've got 4 great speakers today, so I'm really excited. So we're going to start off talking about antibiotic-resistant bacteria, and then move to our phage treatments, and then 2 talks, one focused on Burkholderia caenocepacia, and one on Pseudomonas aeruginosa. So we'll have questions. There's microphones there. We'll also answer questions from the tablet as best we can, so please add those in, and we'll get started. So I am going to start off by introducing Peter Jorth from Cedars-Sinai Medical Center, who's going to talk about new approaches to target antibiotic resistance bacteria in CF. So, okay. So, thank you.
All right. Thank you to the organizers for the invitation to speak today, and thank you all for coming after lunch. Hopefully I'll keep you all awake. So today I'm going to talk about a new project in the lab. Most of it will be unpublished data. I'll start with a little bit of published data that leads into it. But basically we're looking at new ways to target and potentially treat antibiotic-resistant Pseudomonas in cystic fibrosis. I have nothing to disclose. So as this audience is well aware, Pseudomonas aeruginosa infections are still a big problem for people with CF. And it's been known for a very long time that Pseudomonas evolves during these decades-long infections. And the long-standing idea has been that you get these highly virulent, motile, quorum-sensing positive, antibiotic-susceptible bacteria that cause infections early in life. And as the infections progress, these bacteria evolve to become less acutely virulent, form more biofilm by producing large amounts of exopolysaccharides, sometimes lose some of their quorum-sensing systems that regulate these virulence factors, and then often become antibiotic resistant over time. But what we now know is this isn't always true. And so I'm going to give you a couple examples over the next 2 slides. So the first comes from a clinical observational study looking at the development of resistance to the drug aztreonam, which was approved as an inhaled therapy for CF treatment back in 2010. And what they did in this study is they looked at a group of around 300 patients and collected isolates each year over a 5-year period and looked for the development of aztreonam resistance, and then asked, was the incidence of resistance or the arrival of resistance associated with any particular patient outcomes? And a quick summary is that they found that when resistance popped up in these isolates, it was more frequently associated with more common pulmonary exacerbations in these patients. So what this suggested is that antibiotic resistance can arise and these bacteria can still maintain their virulence even with the presence of resistance being there. So, and the way I'm defining virulence in this talk is either increased lethality towards an animal in an infection model or increased cytotoxicity or damage to cells during a cellular infection, which will come up later. So another example came from my work as a postdoc in the Singh lab at Washington. And this is one little bit of a huge study, but I think it encapsulates what I'm trying to show here. So here we have 2 isolates that were collected from an explanted lung. This first isolate is highly resistant to the drug ciprofloxacin and can grow all the way up to this gradient antibiotic disc here, or strip. The second isolate is susceptible to the drug, and you get this zone of clearing around the ciprofloxacin disc, or strip. And when we took these 2 isolates and infected mice, one might have predicted that the susceptible isolate, which is more like an early stage isolate, might have been more virulent to the mice. However, what we saw was completely the opposite. This resistant isolate killed all of the mice in less than 2 days, and the susceptible isolate was virtually avirulent. So this kind of changed the way we are thinking about these infections. It's not just that there's this linear progression towards reduced virulence, but there's actually, the infections are highly complex, and there's actually mixtures of bacteria that are both highly virulent and avirulent at the same time in each individual person with CF. So after this was about the time that I started my lab at Cedars-Sinai, and what my lab is really interested in understanding is why are these antibiotic-resistant Pseudomonas more deadly even in the absence of antibiotic treatment. So the work I'm going to talk about today was all done by Mylène Vaillancourt in my lab, and she's an awesome research associate, and she's presenting a poster, so I suggest you guys check it out to get more details on the study. But basically, what we have here is an intratracheal infection with 2 different strains that were evolved to be resistant to the drug aztreonam. And then we infected mice, and you can see that compared to a wild-type PA01 strain, these 2 aztreonam-resistant strains, like the strain I showed you previously, were more virulent towards the mice and killed them more rapidly than the wild-type. And mutations that we found in these strains in NALD and MEX-R were also found frequently in real CF clinical isolates, suggesting that they're important. And notably, all of this increased virulence was observed in mice that weren't treated with any antibiotics whatsoever. So the next question is, why was this happening? So Mylène focused in on this one isolate that we're calling the C10 isolate. And what we knew is that it had a mutation in this gene called NALD, and this is a transcriptional repressor of the MEX-AB OPRM efflux pump operon, which encodes an efflux pump that effluxes drugs like aztreonam out of the cell. And when you have mutations in NALD, you get increased expression of this operon, increased resistance to aztreonam and related drugs. And so this is shown here in the MIC data at the right. You can see that the wild-type Pseudomonas, when you mutate and become a NALD mutant, you get an increase in aztreonam resistance. And if you delete the efflux pump in either the wild type or the mutant strain, the resistance goes away. And all of this data on this slide and the next couple were just published this year in PLOS Pathogens, if you're interested. Next, we were interested in whether or not this efflux pump overexpression was causing the increased virulence that we saw in this C10 isolate. So we took another approach. Instead of deleting the efflux pump in the mutant, we actually complemented the wild-type NALD gene back. And as predicted, we saw that the resistance phenotype went away by an aztreonam MIC assay. But when we looked at cytotoxicity towards macrophages specifically, we saw that the mutant caused more cytotoxicity in macrophages, but the complement saw no change whatsoever, suggesting that something else was causing this increase in virulence. So we went back and did a lot of searching and head scratching, and what we found is when we resequenced the genome of the C10 mutant, there was one other mutation, just one, and it was in the C-terminal region of this protein called RNase E, which is part of the RNA degradosome in Pseudomonas, and this is an enzyme complex that's involved in RNA processing of messenger RNAs and small non-coding RNAs. And through a lot of work that I'm not going to show you today, basically, Mylène was able to show that this mutation in RNase E was sufficient to increase Pseudomonas damage to macrophages, and it was specifically macrophages, not epithelial cells or neutrophils. And in this cartoon, I'm going to summarize the results a little further. She was able to work out the mechanism. Again, this is all in this PLOS Pathogens paper. But what she found is that RNase E mutations led to increased siderophore production by Pseudomonas, including production of pyocheilin, which, when bound to iron, could induce ferroptosis in the macrophages. And this process promoted increased replication of Pseudomonas and also increased death of the macrophages. And based on this, she tested an idea that perhaps gallium, which looks like iron and mimics iron but is lethal towards bacteria like Pseudomonas, might work as a good treatment for these antibiotic-resistant bacteria. And sure enough, when she treated the C10 mutant or wild type with gallium, it killed both of them and also lowered bacterial burdens and reduced cell damage in this macrophage infection model. So this was all very exciting, but we wanted to take this a step further and say, could we learn something from these virulence mechanisms and find new ways to reduce disease caused by these antibiotic-resistant Pseudomonas? And so our idea came from the finding that ferroptosis was increased in those antibiotic-resistant infected macrophages. And what we saw is that these resistant Pseudomonas increased lipid peroxidation and ferroptosis in the macrophages. And we knew that from other work that ferroptosis inhibitors were available that we could maybe harness to treat these infections. And this is data showing from the Kagan lab at Pittsburgh showing that this clinical isolate, when you infect epithelial cells, will kill them, but if you treat with this ferrostatin-1 ferroptosis inhibitor, it will actually reduce killing of the epithelial cells by the Pseudomonas. So we thought, let's give this a try for our mutant and wild-type infections. So again, we took our bone marrow-derived macrophages and then infected them with different strains of Pseudomonas. We have our wild-type, our C10 mutant, an engineered RNase E mutant over here, and then our complemented C10 mutant. And you can see that all of these strains caused a varying degree of lipid peroxidation and ferroptosis, but with this ferrostatin treatment, you can see in these dash bars that it reduced ferroptosis in all cases. This was great, but it didn't work perfectly well, and there are lots of newer ferroptosis inhibitors that are available, so we decided to test another one. So Milan went out and tested this newer ferroptosis inhibitor called UAMC 3203, and she saw that in this macrophage infection with the C10 mutant, it actually worked a little bit better than the ferrostatin in this preliminary experiment. And so we decided to pursue it further. But before we decided to go any further, we wanted to say, would this even work in a real whole animal infection? The cellular infections were interesting, but what is the real potential for this in an actual infection of an organism? So Milan infected wild type or an ENaC overexpressing mice with Pseudomonas, and then compared lipid peroxidation by 4-H&E staining by immunofluorescence in the uninfected versus infected airways. And you can see that when Pseudomonas infects either wild type or transgenic mice, you get a great increase in lipid peroxidation, suggesting that our ferroptosis inhibitors might be a good tool for us. So the next step was to test this in an acute infection model like we've done before. So in this case, Milan took wild type C57 black 6 mice and then pre-treated them with either this UAMC ferroptosis inhibitor or a vehicle control, infected with our wild-type PaO1, and then measured lung function and bacterial burdens by flex event and CFU counts. And this is what she found in a preliminary experiment, again, a very, very new data. So she saw that bacterial burden was slightly reduced by the ferroptosis inhibitor treatment. And when she looked at lipid peroxidation in the lung tissue by Western blot, she saw that the UAMC reduced lipid peroxidation as predicted. And then when she looked further at the lung function by the flex event analysis, she saw that for a number of parameters, including airway resistance, tissue resistance, elastance, and quasi-static compliance, there were all trends towards improvement in lung function overall with the UAMC treatment. So our next bigger question was, could this actually work during a chronic infection? So again, Milan went back to her mice, but this time, instead of an acute lung infection, she took Pseudomonas, PaO1, embedded them in agar beads, and inoculated them in an infection model that we've taken out to at least 28 days, so we can get good chronic infections from this model. And then instead of pre-treatment, she treated either every other day with the UAMC or a vehicle, or daily. So I'm going to show you on the next slide a summary of data from 2 separate experiments. So here in the closed circles and closed triangles, you can see the data from the every other day treatment, whereas the open circles represent the daily treatment. And right away, hopefully what you see is that there was a slight decrease in bacterial burden by the UAMC treatment. And then there was an improvement in both tissue elastance and FEV 0.1 by the UAMC treatment as well. So this is all, again, new data, things that we need to repeat, but things that we're also very excited about for their potential. So this was all great, but this was all done with a model PaO1 strain. So will this actually work for real-world clinical isolates? And so Milan addressed this question by taking a panel of clinical isolates that we had available to us and first measuring pyocheilin production. Because if you remember from my earlier slide, I showed that pyocheilin is able to induce this lipid peroxidation and ferroptosis in macrophages. So we thought isolates that also produce more pyocheilin might be capable of inducing ferroptosis as well. So she found a varying degree of pyocheilin production by clinical isolates, which is similar to what others have observed before. We have some high producers as well as some low producers and some isolates that produce basically no pyocheilin whatsoever. So she took 4 of these and then decided to do an infection of mice And in an acute lung infection, she measured lipid peroxidation, again, by Western blot. And you can see that the only isolate that really induced a lot of lipid peroxidation was this one that was producing large amounts of pyochilin, also suggesting that these ferroptosis inhibitors might be good for isolates like this particular one. And when she looked a little closer, what was also interesting is that the bacterial burdens were also correlated with their amount of pyochilin production and lipid peroxidation. So she saw again that this isolate that produced the most pyochilin and induced the most lipid peroxidation was able to replicate best during these infections. So to summarize what I've shown you here today, we think that blocking ferroptosis might be a really promising and unique new way to treat these Pseudomonas infections. And this is backed by preliminary data, at least, and trends in macrophage infections showing that we can reduce cell damage in vitro. We can also reduce lipid peroxidation in vivo in both acute and chronic models, and also reduce bacterial lung burdens, improve lung function, and this is, again, in both acute and chronic infections. So going forward, we're going to pursue this full steam ahead. So we're going to test more clinical isolates in vivo and get our N up higher so we can really trust and believe in these data. We're also planning to test this in some of our CF animal models, including this ENaC overexpressing transgenic mouse model that we're using in the lab. And then we're planning to test some other ferroptosis inhibitors that are being developed for treatment of other diseases that might be more readily translatable to humans. So with that, I have to thank my lab, which I'll show you in the next slide, our collaborators at Cedars-Sinai, our statistician who helped us with this, collaborators at National Jewish Health who have helped us on other aspects of the study, Calgary, University of Washington, Caltech, and Pitt, and then our core facilities and our funding. And again, this work was led by Milen. She's at poster 101. She'll be talking about all of this study in more detail, so please check that out. Anna is a postdoc in the lab, and she's talking about her studies of cefidorachal resistance, which are really cool. And Cheryl's another postdoc in the lab, and she's talking about giving a poster about how efflux pump mutations and loss of efflux pump function can increase Pseudomonas virulence. So I encourage you to check all of those out, and thanks to all of them for all their hard work on these projects and related studies.
Questions?
Okay, I'll ask one to start.
Okay.
So can you give people gallium?
They've been trying it in clinical trials, and I know the results have been somewhat mixed. So yeah, that would be potentially a great option. Yeah, yeah.
Do you have a sense like how common the variant that seems responsive is?
Yeah, so we looked at genomes of dozens of clinical isolates, and RNA-Z variants are very common. This specific one that we saw, we don't see all the time, but what's interesting is the mutation that we found was in the part of the gene encoding the C-terminus of the protein, and all of the variants that we detect are in that part of the gene. So it seems like they're being selected for during infection. Yeah. Yeah, in both CF and non-CF isolates, yeah. Yeah.
Hey, Peter.
Hi, Jen.
Great talk, as always. When you look at your evolved isolates, do you see any mutations in LOXA, the lipoxygenase? That was what we saw in that paper.
Right, yeah,
Is there any correlation there?
No, we didn't see that, and that's a great question. We also looked at gene expression, and there was no change in LOXA expression. Yeah,
So
yeah.
different mechanism.
Yeah, so Jen was asking about, there's another gene in Pseudomonas called LOXA, which can also be involved in inducing ferroptosis. And what I didn't show today is that Milen was able to take purified pyochilin and show that that alone could induce ferroptosis in macrophages. So that, yeah,
Thanks.
it's pretty cool. Thanks. Great.
I have a question for you, Peter.
Yeah.
So if you just block CD4 production and that C10 mutant,
Yep.
do you rescue completely the phenotype? Even
Great question. We haven't done that yet, but we assume that would be true. But yeah, we need to test that. Yeah. Great. All right.
Okay. Awesome.
Thank you.
Thank you so much. Okay. I will say also, as I'm pulling this up, we will have some time at the end. So if you think of questions for speakers, we'll have some time that we can ask those of all the speakers at the end. So, okay, so next I'm going to introduce my co-moderator, Daria Van Tine, who's at the University of Pittsburgh, and she's going to be talking about phage therapy for treatment of antibiotic resistance.
Okay, great. Good afternoon, everyone. Thanks, Edith, for co-chairing this session with me. Thank you all for being here. It's really a pleasure for me to be here with all of you and to have the opportunity to tell you about phage therapy and some of the work that's ongoing in my group at the University of Pittsburgh, trying to use phages as a new tool in the antimicrobial toolbox. So as we said at the beginning, I have no relevant disclosures. And today I'm going to tell you about 3 different things. First, I will tell you about phage therapy if you don't already know what it is and how it works. I'm going to tell you specifically about our experience using phage therapy for compassionate use, basically designing precision phage cocktails for individual patients to treat their resistant infections. And then at the end, I'll tell you a little bit about how we've tried to learn whatever we can from the single patients that we treat with phage to try and inform a better approach for the next patient. So, a little overview about phage therapy. So we all know that multi-drug resistant bacterial infections are a looming threat to public health. We know that these infections are still a really big problem for people with CF. And we also, most of us know that bacteriophages are viruses of bacteria, and this means that they can lyse bacterial cells. Phages have basically 2 different options in terms of their life cycle. So once they infect the bacterial cell, they can basically just use that cell as a phage factory and make more of themselves under in the lytic life cycle. Phages can also get into the bacterial cell and then integrate their genomes into the bacterial genome and adopt a chronic or lysogenic life cycle. And when they do this, they basically hitch a ride along with the bacterial host. And it's actually this lytic property of phages, the lytic cycle, that we're interested in when we think about using phages for therapy. So the fact that a phage can kill the bacteria as part of its own natural life cycle for us is a pretty useful property. So we put these 2 things together: increasing rates of antibiotic resistance, decreasing interest in the pharmaceutical industry to develop new antibiotics. This has all prompted a renewed interest in phage therapy. And currently, if you want to access this therapy, you have 2 options. You can find a clinical trial to enroll in, or you can pursue phage therapy on a single patient, single case compassionate use basis. So I recently looked on clinicaltrials.gov and I typed in phage and I typed in cystic fibrosis, and these 3 trials were what came up. So in the United States, there are currently 3 clinical trials for phage therapy for people in CF. 2 of them, however, are not recruiting. One of them is recruiting, and that's this study, the phage study that is sponsored by NIAID and the Antibiotic Resistance Leadership Group, ARLG. And if anybody doesn't already know about this trial and knows somebody who might want to participate in this trial, they are actively enrolling at lots of different sites all over the United States. So there's a contact email, phage@jh.edu, that you can email to get all your questions answered and learn more about this particular trial. So unfortunately, all of these trials, it's great that there are trials. I'll say that at the beginning. It's amazing that there are clinical trials for phage to treat patients. However, all of them are currently for Pseudomonas aeruginosa. There's only 1 trial that's currently recruiting. And all of these trials, understandably so, have pretty strict eligibility criteria. And so for patients that don't have Pseudomonas aeruginosa, that don't live near a trial site, or that don't meet the eligibility criteria for one of the trials, what can they do? So for these patients with drug-resistant infections that might benefit from phage therapy but don't qualify for a clinical trial, one alternative solution is to try and find phages for these patients basically 1 at a time. So this is an individualized precision medicine approach, and we basically are playing matchmaker. So we're trying to match phages with bugs from infected patients. And this is what we do in my lab at the University of Pittsburgh and the University of Pittsburgh Medical Center. So we offer phage therapy for compassionate use in individual patients. And so the way that it works is to do phage therapy, you need phages. And if you want to do personalized medicine phage therapy, you need a lot of phages. So when I started my lab at the University of Pittsburgh in 2018, I set out to do some phage hunting. So I first asked all of my physician colleagues to give me like the baddest of the bad bugs. So I told my physician colleagues, think of a patient that you were unable to treat with the antibiotics at your disposal and give me those isolates. So they did in abundance. And so we used those, what we could say, pathogens of interest for targeting with phage therapy. And we went fishing. So this is a picture of a trainee in my lab named Dan Evans, and he's standing outside a sewer grate. Next to a sewer grate outside a hospital, not our hospital, but another hospital in Pittsburgh. And along with some help from maintenance workers, they've pried open the sewer grate, and that yellow pole is a broomstick handle with a bucket taped to the end of it. And they dip it down into the sewer and scoop up some water. And Dan's holding bottles of wastewater that he then brought back to our lab. And we mixed filtered wastewater with clinical isolates. And a representative plate from that is shown here. So this is bacteria growing in a lawn on the Petri dish. And then we've mixed the bacteria with phage containing wastewater. And those plaques, the zones of clearance on the bacterial plate, are places where phage have gotten in and propagated and replicated themselves by lysing the bacteria. So those individual plaques on the plate represent essentially a colony of phage, where one phage has replicated itself many, many times. So we can pick those plaques and passage them repeatedly to basically isolate individual phages. We can amplify them to make high titer stocks. And the green plate in the middle is an example of a titer plate where we've spotted serial tenfold dilutions of phage onto a lawn of bacteria. And you can see some of the spots that have a lot of phage in them. They totally lyse the bacteria, and other spots that had less phage in them, we can actually see individual plaques. And this is basically how we quantify how much active phage is in a particular sample or a particular lysate. So we can make these high titer stocks of phage, and then once we have a bunch of a single phage, we perform whole genome sequencing to basically QC our process and determine if the phage is suitable for therapy. And that QC process basically looks like, do we have a single phage? Did we manage to actually isolate only one phage? Does the phage genome have any evidence of genes involved in the lysogenic life cycle? So do we think this phage is likely to be obligately lytic, or is it possible that this phage might integrate into the bacterial genome? And then is the phage carrying anything that, as best as we could tell, would be undesirable for putting the phage into the patient? So we look for the absence of identifiable antibiotic resistance genes or toxin genes. So if the phage passes all of those tests, we add it to our library. And the we here is actually many people in my lab, and two of them are shown here. So Sergey Alberdorozko and Nathan Wallace are our phage hunters at the moment, and they're standing next to one of the whiteboards in the lab where we have a phage-o-meter that counts the phage that we have isolated and entered into our libraries. So this slide kind of summarizes 5 years of work by many trainees in the lab, focused on isolating and characterizing phages to build these species-specific phage libraries. So the pathogens that we have libraries for are all listed here, and we're summarizing basically the current totals of the number of phages that we have available in each one of the libraries, and the number of those phages that have actually been used in phage therapy in people. So we have lots and lots of phages for a smattering of different pathogens, and some of those phages have been used in phage therapy. So how does this actually work? Okay, we have phages, we went hunting, we're fishing all the time for phages. So once we have libraries, what can we do? So it starts with a physician who has a patient, and the physician contacts our lab and says, hey, I have this patient. I think they could be a candidate for phage therapy. And if you ask what makes a patient a good candidate for phage therapy, the features that we think about are listed here. So usually the patient has a chronic, either persistent or recurrent infection. They have had prior rounds of antibiotic treatment that they have failed. So the prior antibiotic treatment is either ineffective or effective options for treating the patient going forward are just not possible for whatever reason. We think the patient might benefit from phage therapy. And importantly, also, the patient has a long enough time to wait. We have to identify phage. If we find phage and the patient wants to pursue therapy, we have to manufacture phage for them. We have to formulate the phage. We have to test the phage, and then we have to ask FDA for approval to treat the patient. It's a very long process, can take weeks to months to do. So a patient who is critically ill and in dire need of phage is usually not a good candidate for phage therapy, unfortunately. So this is how we identify patients. Once we identify a patient as a candidate, their isolates come to our lab at the University of Pittsburgh. We perform susceptibility testing, and we test susceptibility and report the results back. So an example of the kind of susceptibility screening report that we generate is shown here. So we're summarizing the species, any clinical notes, things we know about the patient, the person that screened it, which in this case was Sergey, and then the results of the screening. So this picture on the bottom right of the screen is showing basically a spot test where we take our individual phages in our library and we spot the lysate of each phage onto a top agar lawn of the bacteria. And where you see zones of clearance, those represent activity in the lysate. And this initial screen gets followed up by a titer where we actually confirm that the inhibition, the activity we see, is due to phage. And in doing this screening, we identify phages that look like they have greatest activity against the isolate that we screened. And so we give this information back to the physicians, and then the patient and the physician decide whether they want to pursue phage therapy or not. So this, to summarize sort of everything that we've done to date, we have received almost 120 requests for phage susceptibility testing from 120 different patients. 61% of the time we had phage that were active. So for over half of the patients, we actually had phage in our library that were active. That boils down to 15 patients so far that have been treated with phage. And so the attrition rate here is quite large, but still we started with 120 and we ended with 15 patients so far that have been treated with phage. So who are these patients? What sorts of conditions do they have? So the clinical conditions for all the patients are summarized here. About a third of the patients are CF patients. A third of them are split roughly between people with enterococcal infections, whether it's a recurrent bacteremia or a prosthetic joint infection with enterococcus, or a lot of requests come from patients with left ventricular assistive devices or LVADs that are chronically infected. And then the remaining third are a smattering of other conditions. And then what are the bugs? Well, what we screen for is what we have phage for. So about half of the organisms that we've screened have been Pseudomonas aeruginosa. That's the most popular request. And then the other half of the requests have been really a wide variety of different organisms. And somewhat not too surprisingly, but importantly, the likelihood that we can find active phage is pretty variable by pathogen. So for some pathogens like Pseudomonas aeruginosa, almost 75% of the time, we have phage in our library that are active against the isolate that we screen. And that ratio is just about flipped for Burkholderia. So only about 25% of the time, 30% of the time do we have active phage against the Burkholderia that we screen. And Enterococcus is split right down the middle. It's like a coin toss at the moment, whether we have phage that are active or not. Okay, so we found phage, the phage are active, the patient and the physician want to proceed with phage therapy. What happens next? So we have to make phage for the patient. And what we often do is use the patient's own bacterial isolate to manufacture the phage. And this keeps the phage trained on the bug we want to target. From a regulatory perspective, it's also more permissible to use the patient's own isolate as opposed to a different host to propagate the phage. So we make a bunch of phage, we purify, we formulate, and then we undertake a series of regulatory steps with both the FDA and the local IRB, either at our institution or at the institution where the patient's being treated. And these regulatory steps and all of the testing we do are focused on trying to ensure that the phage is active on the patient's own isolate, that the preparation is free or has very low amounts of endotoxin and exotoxin, and also that the preparation is sterile. And we have to do this kind of testing on every single phage and every cocktail that we make for a patient, because again, this is individual, one patient at a time, compassionate use phage therapy. So once we do all of that, we get permission to administer phage to the patient, and then importantly, we monitor them to make sure that they don't have any adverse reactions, that they can tolerate the phage therapy, and then we monitor over time, right, for clinical response. We look for development of phage resistance, and we also look to see if the patient is mounting an antibody response to therapy. And so since 2019, we've treated 15 patients, both at our center and elsewhere, with phage for multi-drug resistant infections. And I want to acknowledge all of the amazing people that help do this work. So Gadi Haidar, Madison Stelfox, Ryan Shields, they're all clinicians at UPMC. Kaylee Hughes-Kramer in the middle handles all of the regulatory work and liaising with the FDA. And then, as I mentioned before, Nathan Wallace-Sergey are basically our phage factory in the lab that actually make the phage for all of the patients. So this is a very busy table, but it summarizes all 15 of the patients that we've treated. So 3 of these patients had cystic fibrosis. They were infected by a variety of different pathogens, a lot of Pseudomonas. They received a bunch of different phages by a bunch of different routes. And the outcomes were also variable. And I'm actually going to go to the next slide, which dropped off the final 2 patients because we don't have enough time to assess outcome with those patients. And the 13 where we've assessed an outcome are listed here. The patients highlighted in blue are the patients where there was no discernible response to therapy, usually because the patient received therapy for a very short amount of time. The other 9 patients highlighted in green are patients where we observed them for long enough that there was a detectable response, either microbiologic or clinical, and that it looked like it was a positive response to therapy. Very importantly, phages were safe in all patients. No adverse effects, no every patient tolerated phage very well. There was no discernible, it was actually kind of boring giving patients phage because we kind of are bracing for a dramatic response that the patient is going to have an adverse reaction to phage, and that has not yet happened. Hopefully, knock on wood, that phages remain safe in all these patients that get them. So this is heterogeneous, right? There's a lot going on here in a table like this because, again, this is personalized. This is individual, compassionate use phage therapy. But I'm a researcher. I run a research lab. I want to help patients by making them, custom cocktails of phage therapy, but I also want to understand what the heck is going on when we give a patient phage therapy. So in the last part of the talk, I'm going to take you through sort of an in-depth study of one patient. It's the first patient on the list, patient number 1. This was a 32-year-old female with cystic fibrosis. She was known to be colonized with Burkholderia multivarians since at least 2001. She had a double lung transplant in 2017, and we started following her in the fall of 2020 when she had pneumonia and sepsis and was in the hospital with multi-drug resistant infection with this Burkholderia multivarians. So this study is published in Transplant Infectious Disease. I'm just going to take you through some of the salient findings from this study. So this kind of timeline is summarizing the treatment during the time that we followed this patient. So we followed her for about a month before she had phage therapy, and then about a week that she received phage before, unfortunately, she passed away from her infection. The blue shaded bars are showing all the antibiotic therapy that she received. And above the timeline, the green and red boxes are showing her cultures. And those red boxes that say BCC are cultures that grew Burkholderia multivarians. So you can see that despite all of these antibiotics, her cultures remained positive with Burkholderia multivarians. The phage search for her kicked off, maybe halfway through the pre-phage period, and in only 10 days, our colleague Ben Chan over at Yale was able to identify a phage, make phage for this patient, and then get it to Pittsburgh to treat the patient. So this patient received inhaled phage for 8 days and a single dose of IV phage before she ultimately, unfortunately, succumbed to her infection. So from this patient that was unfortunately a clinical failure of phage therapy, we wanted to try and understand more about what insights could we take from this case to try and do a better job for the next patient. So during the time that this patient was receiving phage, we were able to sample her airway. So we received airway specimens, either sputum or tracheal aspirate. And from those airway specimens, we used quantitative PCR to try and quantify the change in bacterial abundance and also phage abundance over time while she was receiving phage therapy. So the B multivarians abundance is shown on the left, and we can see that after initiation of phage therapy, there was a small but significant decrease in the bacterial abundance in these specimens. And then the middle panel is showing the phage abundance. And surprisingly, for the first 5 days of therapy, we didn't detect any phage using qPCR in the respiratory specimens. However, on day 6, the phage popped up. And we don't know if this is because it took this long for the phage to find its host and replicate. We wonder about the dynamics of phage replication in a host. But nonetheless, after 6 days of phage therapy, we detected phage in her respiratory specimens. And then we also looked for evidence of serum neutralization. So after a week, we wouldn't expect a neutralizing antibody response, especially in a transplant recipient that is immunosuppressed. But we nonetheless looked and didn't find any evidence of serum neutralization. So I mentioned that this patient had been colonized with Burkholderia multivarians since 2001. So John LaPuma in Michigan provided us with historic isolates sampled from the patient early on during her initial, when she was initially found to be colonized with Burkholderia multivarians. So we collected all the isolates that we could from her over time, sequenced their genomes, and found that, as we expected, the isolates were closely related to one another, suggesting that she had been colonized for a very long time with this organism. And then we also looked at the antibiotic susceptibility and also phage susceptibility of all of these isolates from this one patient. So the isolates are shown in the rows on this matrix. Each row is a different isolate. They're shaded based on the date of isolation, and then their antibiotic susceptibility or phage susceptibility is shown in blue for sensitive, yellow for resistant, and the phage that she was treated with, BCH7, is shown all the way to the right of the matrix. And what we see is that early on, her early isolates were relatively susceptible to antibiotic. They were actually resistant to the phage that was used for therapy. Her later isolates that were more antibiotic resistant were actually phage susceptible. And we confirmed this, right? We can show this using titering. So an early isolate, the very first isolate we had from her from 2001, resistant to phage. However, a later isolate was now phage susceptible. And through whole genome sequencing and some LPS analysis, we confirmed that this later isolate actually lacks O antigen, and we suspect that this maybe had been masking the receptor that the phage is using for infection. It raises interesting questions about when is the right time to look for phage for a patient, but nonetheless, it suggests that banking isolates from patients over time and testing their phage susceptibility could give you a leg up on using phage to treat them in the future. So with that, I will wrap up. My conclusions from all of this are that phage therapy is available, but access is still very limited. While trials are underway, many patients don't qualify for them. And for those patients that want to access phage therapy, they do have another option, which is seeking out compassionate use. However, this obviously happens at a very small scale. It's very labor intensive, but we can do it. We have in our lab phages for lots of different patients, but we have to play matchmaker and match phage for each patient individually. So there were 13 patients that we've treated and assessed outcome. 70% of those patients experienced some kind of improvement in their clinical course. The other 4 patients had no response, and importantly, none were harmed by phage therapy. So we're participating in and planning clinical trials of phage therapy, hoping to grow this technology and this treatment modality. And one in particular that I'll highlight is we're planning a phase 1 pilot trial of Achromobacter, a fixed cocktail of 5 Achromobacter targeting phages. We're hoping to enroll 10 participants with Achromobacter colonization at 2 sites starting hopefully in the next year. And I want to wrap up by thanking all the people in my lab. They did all this work that I get to come here and tell you about today. I'm grateful to our funders. I thank you for your attention, and I'm happy to take questions.
That was great. Thanks. Not on. That was great. Thank you. Yes, question.
Great talk.
Thanks.
I have it occurred to me that phages are really good at moving genetic material around. Do you have any information for any of your phages about the ability to, for instance, move an antimicrobial resistant plasmid from one organism to another? And then a second question is, how long do the phages hang out after you've treated with them?
Yeah, those are fabulous questions. Okay, so phage transduction is the ability of a phage to package DNA that's around its environment when it's making itself, and to move that DNA into its next host. So we have not assessed transducing potential. We've been asked before about transduction potential of our phages. When we make phage using the patient's own isolate, we think that this risk is minimized because whatever DNA might be delivered to the bacteria in the patient would have originated in a bacterial isolate from that patient. So this genetic system is closed. It's a valid question. It can be difficult to assess, but it's definitely something that we should be thinking about. Yeah, and your second question was about, remind me of your second question. How long do they stick around? Yes, another fabulous question. We don't know. So phage kind of break the mold when you think about antimicrobial pharmacokinetics and pharmacodynamics. They are self-replicating. They're self-dosing. If they find suitable hosts, they're also self-limiting. When their hosts are gone, they can't replicate anymore. So the traditional kind of models for pharmacokinetics, pharmacodynamics probably won't apply to phage. So the patients that we treat at UPMC, we sample them pretty intensely so that we can assess pharmacokinetics, pharmacodynamics, and that's work that's kind of going on at the moment in our lab to try and understand, after we give a patient phage, how long does it hang around, where in their body does it go, to try and answer some of those questions. Yes.
Great talk. You kind of answered some of my question, part of my question. I guess the unanswered part is, how do you determine route of administration and length of treatment?
That's a great question. Okay, route of administration, I'll go back to the crazy table, and we can meditate, we can look at, so this is personalized. We try to optimize for the patient, and we try and deliver phage to where we think the bug is. We have favored systemic, so IV administration, to basically try and put phage everywhere, and then also tried to add one additional route or more if we think we need more. So for patients with pneumonia, with a lung infection, those patients get IV phage and also inhaled phage. We recently have had patients that have we know that they have bacteria in their throat, and in one patient where they have a cuffed trach, that their upper and lower airways are separated from one another, we know they have phage in their throat, we know they have phage in their lung. They've gotten inhaled phage through their trach, and then we've also been spraying with an atomizer phage into their mouth and throat to try and basically put the phage where we think the bacteria are. For a patient with enterococcal infection, where we knew the patient was colonized in their GI tract with enterococcus, that patient got oral phage therapy in addition to IV. So we try and do IV, it's like IV plus one, but for each patient, we have been trying to optimize the route. For frequency, dose, and duration, so frequency is once a day, twice a day, whatever is feasible for the patient. Dose is the highest concentration of phage that we can give to the patient, given how much phage we have and how much endotoxin is in the preparation. And then duration is we try and treat for 4 to 6 weeks because many of these patients are quite ill. We don't know what the optimum treatment course should look like. I will say, our physician colleagues, so given the great safety profile of phage, they basically come back to us and say, well, why not keep going? And so I find myself now asking the physicians, do you have to keep treating? Can we take a break and see what happens? Given the great safety profile and the lack of side effects, some of our patients have been on phage for multiple months for their duration.
I have a couple of online questions. You sort of answered some of this, but are you only giving this at Pittsburgh, so they have to be?
So half the patients that we've treated have been at UPMC. The other half have actually been elsewhere. And so we manufacture the phage in my lab. We do all the testing in my lab, and then we actually ship the phage to other sites, always to an investigational pharmacy. So we don't ship phage to patients at their homes. We ship phages to institutions, hospitals, usually academic medical centers that have investigational drug services that can receive and then dispense phage for therapy.
And then someone asked about the side effects. Do you do any pre-treatment, like albuterol or anything like that, when you're doing inhaled phage?
We have not. We have been, for patients that get respiratory therapy, we usually have that be separate from the phage therapy to avoid combining things and having unexpected things happen. For the patients, a lot of patients that we've treated in the hospital, it's coordinating with the respiratory therapists, the patients that get inhaled phage. But again, for every patient under like an individual protocol, basically anything is possible. And it often feels to me like we're building the airplane just as we're flying it, because we get asked questions like this about what should we do for this, what should we do for that? And we say, well, what makes sense for you or for the patient? We really have no guidelines in this area at the moment.
Questions in the room? I can keep going.
How about one more, and then we'll move on.
Yeah. So what they asked, does phage therapy have any impact on future drug resistance or acquisition of other pathogens, and any impact on lung transplant eligibility?
Yeah, we have no idea. So our transplant infectious disease physicians that evaluate patients for transplant are now considering phage as a potential decolonization or right after transplant as a potential tool to use. So we've established a strong working relationship with the transplant infectious disease team, at least at Pitt, to get them to think about phage when they're evaluating patients for transplant.
Awesome. And regarding like emergence of other pathogens, have you seen?
Yeah, we don't know.
Okay.
Yeah, it's very early days.
Can patients from Australia access this?
So Australia has its own phage therapy network. It's called Phage Australia. So don't come to me all the way in Pittsburgh. They can go to there are centers in Australia that they could access for phage therapy.
Awesome.
And it looks like Sage has a question.
I think
Yeah.
one more.
Okay, one.
I just wanted to know about how you consider antibiotic use and that sort of stuff in your phage hunting and in your design.
Yeah,
for
so
these personalized cases.
yeah, we get this question a lot. So at the moment, we're not set up to do rigorous like synergy testing, susceptibility testing alongside antibiotics, and I'm not sure. Well, so we also don't know about correlates of like if we find synergy in vitro, we're not sure if that's going to translate to synergy in vivo. Given all the different moving parts, what we tell the physicians is whatever antibiotic therapy you would already be giving this patient, keep giving it, and phage is adjunctive. It's just an add-on to whatever the patient would receive if phage weren't available. But as we do this more and more, we know what antibiotics these patients got. We can go back into the lab and test them. And hopefully in the future, we could be in a position where we say, if you give this phage, it should be in partnership with this antibiotic. Yeah. Okay. I think unfortunately we're going to move on. I'll be around after the session, and we have time at the end of all the talks for questions. But I'm going to invite up the next speaker now. So our next speaker is Sylvia Cardona. She's a professor at the University of Manitoba. She's going to tell us about machine learning, chemogenomics, and Burkholderia cenosepacea. Sylvia, welcome.
Thank you, Daria and Edith. Thank you, everyone, for being here. So,
You might have to click on it.
oh, I see. Awesome.
I think it should work now.
Yeah. Yeah, I'm finding the laser pointer. Okay.
Yeah, I think it should work.
Yeah, thank you. No relationships to disclose. So I'm going to start by showing this diagram about polymicrobial infections in people living with cystic fibrosis, and we all know about them. There's a cycle of inflammation, infection, treatment with antibiotics, and from this cycle, I like to point out that 5 to 30% of people with cystic fibrosis get infected with Burkholderia cepacia complex. And as we all know, Burkholderia cepacia complex is one of the most resistant bacterial pathogens that are known. They are inherently resistant, and they become more resistant with antibiotic treatment. 70% of these individuals become chronically colonized, and unfortunately, their outcome is clinical deterioration and sometimes fatal pneumonia, which leads to dysphagia syndrome, as you all know. So it's obvious that new and more effective antibiotics treatments are needed to treat dysphagia infections. So I started many years ago working with Burkholderia and developing lots of genetic tools to understand its pathogenesis. And I got engaged in trying to help patients and finding new antibiotics. So I started with screens, classical screens of antibiotic molecules that are, many people know, working with small molecule libraries, assessing these small molecules for antimicrobial activity, for growth inhibitory activity, and finding small molecules that would inhibit growth. And in antibacterial screening, we call these molecules hits, the ones that are active. So from 30,000 compounds that I screened this year, I found around 200 hits. And when we start to do the data mining, filtering out the ones that were known antibiotics or antiseptics, or they had already known toxicity, you know, the list went smaller and smaller, and at the end, we end up with compounds with very low efficacy, low solubility, or they were toxic. So basically, I have to say that we failed in finding anything that was interesting to follow up. So this is common in early antibiotic discovery, and you can see here a timeline of what we do with when we work in antimicrobial discovery. We start with the primary screening, which has a high attrition rate, less than 1% of the molecules screened. You can call them hits. Then the hit to lead steps, with what is finding out the mechanism of action, looking at toxicity, also has a high attrition rate. Less than 1% of the molecules that you first found pass this step. And this goes on and on for every step until going into preclinical studies, you have less than 0.5% of the molecules from the previous set. So you can imagine that if you multiply the probabilities, this is what you see. You have to start with more than 10 million molecules, where if you are lucky, you are going to have one at the end. This takes many years, and of course is resourceful. It takes a lot of money to come back with a new antibiotic. Not surprising that we have new ideas now, like the ones developed by Peter, by Deria, looking for solutions. Because when you work with in antimicrobial discovery, you pretty much feel like this little monkey. So it's such a huge task. So you really feel that you're catching this big elephant. So what can we do? I love the talks that I've seen before, because they're all trying to find solutions. In my case, I found help with artificial intelligence. But when I say artificial intelligence, don't get me wrong, I mean human intelligence. So that were my colleagues, Rebecca Davis, a chemoinformatician, and Pinsao Hu, an expert in computer science and machine learning, that came to the rescue. And I'd like to give you a bit of a background in artificial intelligence, because people are talking about it. So artificial intelligence is, in a way, making computers do things that humans do. And actually, you teach the computers to do that things, but hopefully things that humans find boring to do. Now, if we get a step further and we talk about machine learning, it's about math, it's about algorithms that learn with data and provide outcomes. Now, the next generation of machine learning is deep learning. And deep learning is great to work with complex data sets, as the one we deal with when we work in drug discovery. Deep learning is able to has different different layers of learning, and that's why it can process data that is complex. Also, machine learning or deep learning is all about data. You have to have data. And in the case of drug discovery or antimicrobial discovery, you usually have chemical compounds with very complex structures, thousands of them, and then you have activity data. So those are very complex data sets that deep learning approaches can handle. And then what you are expecting is that the models will learn from the data provided and will produce outcomes. And the outcomes will be predicting what could happen in the future. So the question we asked with my colleagues was, can we predict antimicrobial activity in small molecules, in chemical molecules, only knowing the chemical structure? That was the question that we asked. As I told you before, when you work with deep learning, you work with data. So what do we do with data? This is a typical set of sets that we use in machine learning. First, you need a data set. The data set is divided in training, validation, and test sets. So as you see in the diagram there, we have 80% of the data set goes to training and 10% goes to validation and training. So the models are developed with a training set, and then they are perfected with the validation set, and finally are tested with a test set that hopefully, ideally, has the model has never seen the test set. This is crucial to get good evaluation of the model. So then the model is evaluated and a performance of the model comes up, and you will see different parameters like the ROC or the precision recall curves. You also will see accuracy. All of them, you have to see how close they are to 1. That's the point. So once predictions are made with the data sets, you would like to hopefully take them to the real world, and that's where experimental confirmation of those predictions come in place. So that's what we did, and I'm showing here the general diagram, and Chen Yu Liu, a student with Pinsao Hu that worked implementing the model. So our training set was the screening, that failed screening of 30,000 compounds in a species of Burkholderia cepacia complex, Burkholderia cynocepacia. So we got these 30,000 compounds where we knew which ones were active and which ones were inactive, and binarized the data. So we call them either active or inactive with numbers, and then we describe the molecules in a way that a computer can understand, bits and counts. And in this case we use, and this is where the deep learning is applied, we use a model that is called directed message passage neural network. And basically what the model does is describing the three-dimensional structures of the molecules from atom passing through the bonds to the next atoms, and building some kind of a three-dimensional structure of the molecule in vectors that the computer can incorporate. So the model was trained with this data, and once the model was trained and achieved a good performance, then we asked the model to predict activity in 2 virtual compound libraries. One was the FDA-approved compounds, and the other is a library of natural products, the zinc library. Of course, these were virtual libraries. We had the chemical structures, and they were described in the same way that the model was trained. And then we asked which of these molecules were predicted to be active and which ones were predicted to be inactive. So once we got the list of potentially active molecules, we purchased about 50 to 100 molecules for each of the libraries, and we tested them in the lab. That's important, the experimental confirmation. So And we tested them in the same way that the data had been produced before, calling actives the one that had more than 20% growth inhibition. So these are the results for the FDA-approved library, and you can compare here the hit rate. The original hit rate, less than 1% for the 30,000 compounds that we screened, and the experimental screen that comes from the compounds that were predicted to be active by the deep learning algorithm, the hit rate was 26%. This is about 30-fold increase in the hit rate. For the natural product library, the hit rate was a little lower, 12%, but still 14-fold increase from the previous screen. So we were excited. My colleagues in computer science were not. They couldn't understand why the high prediction that they got from the test set, 90%, didn't translate into the lab. And I told them, welcome to biology. So Jisan, a student in my lab, was in charge of the wet part of this project. So one compound that we got from this experimental confirmation of predictions was this one that got our attention because it's totally different from any known antimicrobial that is available. So immediately the question is, how does this guy inhibits growth? And that's the second part of what we do, is finding the mechanism of actions of novel antimicrobials. And when I talk about mechanism of actions, I talk about two things: the drug binding the target, inhibiting, corrupting the target, and everything that comes after as a domino cascade. All the secondary effects that are triggered by the antibiotic. And we do that by exploring chemical genetic interactions at the genome level. We try to, we build thousands of mutant strains, and by looking at specific hypersusceptibility of these mutants against the drugs, we kind of make hypotheses about possible mechanism of actions. These mutants can harbor mutations in the target or in genetic elements related to the secondary effects. So the libraries are created in my lab by awesome people like Jisan, Jules, Andrew, and Dustin. And we have currently two types of libraries in Burkholderia cynocepacia. One is a library of essential genes, that is mutants that have CRISPR technology, which allow us to silence essential genes to assess the phenotypes. And what is nice about the CRISPR interference or CRISPR-I libraries is that because of course, CRISPR technology includes working with guide RNA sequences. Each of these silencers of gene expression are unique to each gene, so they can be used as unique DNA barcodes to track the mutants, as I will explain in a minute. So the other libraries are transposon mutant libraries of all the other non-essential genes in Burkholderia cynocepacia, and with the transposon we deliver again a unique DNA barcode that allow us to track the mutants. And the way we do it, I'm showing this scheme to simplify the process. All the mutants are grown in a pool together in a test tube, and then the pool of libraries are grown in different, exposed to different compounds, the antimicrobial molecules we want to address the mechanism of action, and of course we compare them with the control. And because all the mutants have these unique DNA barcodes, is that we can track them by Illumina sequencing of the barcodes, and then counting the number of barcodes that we find for each mutant, we can estimate the relative abundance of these mutants in the population. Also, because the data is complex, we have different ways to look at the data. For example, heat maps, or scatter plots, or volcano plots. And to make it simple, the mutants that are hypersusceptible have high gene fitness in in the control, but low gene fitness in the antibiotic experiment. Same with the Vulcano plot, we can find the fold changes in fitness, and the ones that have negative fold change are the ones that are hyper-susceptible, and Vulcano plots also provide the statistical significance of the fold change. So going back to my compound that we found thanks to artificial intelligence, we did a CRISPR-I-seq analysis of this compound, and we found that 2 mutants were specifically hypersusceptible against this compound, and both of them, a peptidyl tRNA hydrolase and a ribosomal protein, tell us that this compound is targeting protein synthesis. Also, clonal assays looking at hypersusceptibility at different concentrations of the drug tell us that this is most likely the case. So moving forward, you want to do things that are also closer to the patients, because even when you find a new antibiotic, taking it to the market can take years, right? Because of all the processes that are involved. So one approach is to enhance the activity of already known antibiotics, and that's what we did with exploring the vulnerability to beta-lactams, for example. So I'm going to show a work by Andrew Hogan, who explored about 20 cell-envelope antibiotics, including beta-lactams. And what he found is that all the mutants that were hypersusceptible to beta-lactams were in processes related to glycan synthesis. And to put it short, we found that there was a common intermediate in all these processes, that is andecaprinol phosphate. Every time we hit mutants that have andecaprinol phosphate synthesis or recycling disturbed, then we see beta-lactam hypersusceptibility. So we think we have the Achilles heel of beta-lactam susceptibility for Burkholderia cynocepacia. We also went further and looked at other potentiators of beta-lactam susceptibility, like, for example, the beta-lactamase inhibitors. This is the case of avibactam, a very well-known beta-lactamase inhibitor, and we found the target of avibactam by looking at this hypersusceptibility of different mutants. As you can see here, PEN-B had susceptibility of all these antibiotics, and when we went further and looked at the susceptibility of the PEN-B mutant to Astreonam, ceftazidime, and meropenem, we found that the mutants were, of course, more sensitive to these antibiotics. But then when we added avibactam did nothing to the susceptibility because the beta-lactamase was simply not there. So we found that PEN-B is the beta-lactamase that is targeted by avibactam. But taking this to the clinics, we wanted to see if these combinations, how they will work in clinical isolates. So as I'm showing you here, these graphs show several clinical isolates of Burkholderia cynocepacia from Canada, and you can see that they are a graph based on the percent of mutants with percent of clinical isolates with MIC, and these graphs show that the closer the lines to the left, that means that more mutants have lower MIC, lower minimal inhibitory concentration to that antibiotic. And you can see that when we add avibactam for Astreonam, for meropenem, and ceftazidime, you can see that because of the shift of the curves, that avibactam potentiates more strongly Astreonam and meropenem than the classical combination with ceftazidime. So we can we think that this is a way that, you know, by performing these chemical genomic experiments, we can produce this kind of data that physicians can look at and plan clinical trials with different combinations. So I'm going to finish by summarizing my work. Artificial intelligence increased the hit rate of early antibiotic discovery, but at least 14-fold, which is exciting. We are continuing working with this. We found a novel growth inhibitor that was experimentally confirmed, and CRISPR-Cas9 suggests that it's an inhibitor of protein synthesis. And lastly, chemogenomics identified new targets and new drug combinations for enhancing the action of beta-lactams against B. cepacia complex. So I'm going to finish by thanking people in my lab and the funding. Happy to answer questions.
Questions? Yeah, thank you very much. It's very interesting.
Thank you.
Great.
Okay, I'll start with a question. So
Okay.
in your mutant screens, it seems you focus on mutants that are hypersusceptible. Do you ever encounter mutants that are more resistant?
Yes, so it's common to find both, and we have another work going on looking at those mutants that are resistant. Yes.
Great talk. Have you tested those molecules on animal models and see if they're super efficient too?
Yes, no, we haven't done it yet, but of course, any molecule that is enough interesting, we would like to do those things. This particular growth inhibitor that I shown is a weak growth inhibitor, so we will have to find the target, chemically modified, see if we can increase efficacy to merit, you know, going further. Yes.
Thank you for the interesting talk. Since your molecule is potentially targeting protein synthesis, do you expect some known ways to escape protein synthesis inhibitor would also escape the killing of this molecule?
So your question is about that the growth inhibitor that inhibits protein synthesis, right? And whether I expect additional resistance?
Yeah, like the known ways to resist to like currently known antibiotics to then target protein synthesis would do.
Yes. Yes, very interesting question, and it takes me to the idea of that antibiotics of novel classes shouldn't have cross-resistance with known antibiotics, right? So, for example, if this is a known antimicrobial molecule that inhibits protein synthesis, will other antibiotics that also inhibit protein synthesis, mutants that are resistant to that, will be resistant to this one? It could be depending on the target and the type of mutation, right? An idea that brings optimism is that antibiotics totally new classes shouldn't show cross-resistance with old antibiotics. However, we don't know if that is true. For example, if the mutation is in an overexpression of efflux pumps, and efflux pumps can be very promiscuous, right? Maybe they can efflux the new antibiotic as well. We hope that this will be the case, and there are examples of new antimicrobial drugs that don't show cross-resistance. We don't know if it shouldn't be the case for all of them. We have to analyze each case. Yeah, thank you.
Okay, let's thank Sylvia again.
Thank you.
Okay, so our final speaker in the session is Dao Nguyen. Dao is an associate professor at McGill University, and she will be talking to us about new therapeutic approaches to treat Pseudomonas.
Thank you very much for the invitation to give you the last talk of this afternoon. Hopefully, I'll hold your attention until the end. So I was first asked to talk about new emergent therapies, and I was asked by the chair to give a little bit of a overview of the field, which was really a daunting task because there are so many ideas and so many preclinical compounds that have been tested, et cetera. But hopefully, I'm going to give you Dow's perspective on the new emergent therapy. So I apologize first and foremost if I have missed your favorite pathway or ideas, but I will explain to you, what the thought process into my choices are. It's not. Oh, this one.
No, you can use that. It just has to click first, and then now it should work. Yeah.
Okay.
Now you can use it.
Okay, so the disclosure, so I do sprinkle at the end a little bit of our own work in the therapeutic space, which is just a limited amount of what my lab does. Related to that, we are using compounds that are provided as in-kind reagents by AstraZeneca, and otherwise the work has been funded by the CF Foundation, CF Canada, and Glycan in Canada. So, there's been decades of work looking to understand the mechanism of why does Pseudomonas persist in CF, and why does it respond poorly to antibiotics. And the mechanisms are varied, and I could probably, spend hours trying to review that literature. But needless to say, some of the major themes that have come across is, well, in the context of the CF lung environment, there are very specific features of the chemical, physical properties of the CF mucus that Pseudomonas is able to grow as a biofilm, that are many mechanisms that make it tolerant to antibiotics or develop genotypic resistance. These are polymicrobial interactions. And on top of that, add the layer of diversity, both phenotypic and genotypic diversity, and the fact that it can resist defenses from the host immune systems. So there are many, many different roads. And so of those, which ideas, which concepts have actually led to some emergent therapies that are close enough to the clinic now? And so before I went to some of the emergent, new ideas, I wanted to give you just a glimpse of what is actually in the clinical pipeline. And so what this is the clinical pipeline of compounds that have been, in the last 5 years, been in either somewhere between phase 1 and phase 3 clinical trial. And I'm going to outline to you what has been cooking against for Pseudomonas infections. So what you can, I'm not going to go through every single of them. In fact, I probably do not know, in personal, intimate detail, the history of every single compound I'm going to show you. But this is really just to give you a glimpse. And the glimpse that you see is that first and foremost, most of the compounds in the pipeline are traditional antibiotics. So there are a variety of different cephalosporins, carbapenems that are all being tested with, the latest snazziest beta-lactamase inhibitor. So that's really the bulk of the pipeline. There's a few novel targets or mechanism of action, which include some new LPS pathway enzymes like LPTD or LPX inhibitors. There's some that are energetic inhibitors like bismuth ethane diethyl, or things like antibiotic that are coupled with siderophores. Then we move on to a few polymyxins, which are polymyxin derivatives, and these are really primarily used as permeabilizers with the idea that they could be used in conjunction with an antibiotic because now you have made the outer membrane more permeable. And then there's others, and in here you'll see a number of compounds that I'll talk in more detail, such as those that impact iron metabolism. Phage, which I won't cover, but has been beautifully covered previously, and things like NO, which is still on the pipeline after several decades of study. And then in a more, if of those, which are the ones that are targeted for CF, so they're just highlighted here in yellow. And then after 5 years, so this was what was starting 5 years ago. Hang on, I'm trying to move this. Okay, there you go. Okay. And then since then, where are we at? So everything that's grayed out has dropped out of the pipeline, and there has been 2 compounds that has been approved, and they're basically standard antibiotic. But then quite a few of things have been kind of abandoned, kind of midway in early phase clinical trials. So again, those are all the grayed ones. If we move to some sort of less conventional approaches, there's some antibiofilm compounds here, and I've highlighted a few that are sort of not quite mucilic, but will disrupt the matrix through, for example, alginate oligosaccharides. Some of them are novel antibodies that will sort of bind a human protein, a human antibody actually that will bind some proteins that are important to the structure of the biofilm matrix, like the anti-DNA B2, which is also a novel target. And then in there, things that you may not think about are a passive immunotherapy, so using monoclonal antibodies of a variety of flavors to try to enhance opsonophagocytic killing or neutralize a virulence factor. And then there's a lonely antivirulence, a small molecule compound. Of those, several of them antibiofilm compounds are targeting the CF. And then again, in fast forward 5 years where we are now, none of them have been approved, and then quite a few of them have been abandoned as well in this pipeline. So the take home is that there are really not many candidates that are reaching phase 3. And so, Sylvia Cardona definitely outlined how challenging traditional antibiotic discovery is. The non-conventional ones are also incredibly arduous. What we have right now are really just 2 successes, which are variation of antibiotics that we know. And there's really just one new target that has only one drug that has a new target, and then it has failed in phase 3 because of toxicity. Most of these really remain in very early clinical phase, and then there has been yet no track record for successful development of an antivirulence, antibiofilm treatment, despite the fact that we've been studying these targets and saying they would be great new therapies for decades now. So it's a little, humbling. And a number of these drugs are also pivoting to inhaled formulation because they're turning out to be too toxic or too challenging to formulate systemically. And also there's no vaccine. I think the vaccine world has given up on Pseudomonas, at least at this point. So to go back to these mechanisms, what I wanted to now give you is a few sort of short stories. Most of them are work by others, so I hope I do them the due respect they deserve. And what I decided to choose were a few pathways that tell us something about the specificity of Pseudomonas within the CF lung environment, and that have led to work that has advanced enough in preclinical and early phase clinical trials. So these are actually things that are being tested in humans, at least, because those that are even compounds that have made it to mouse but not even humans litter the literature, and many of them never go beyond the mouse study. So if you ask, most Pseudomonas microbiologists, they will tell you that they can disagree on many things, but they will all agree that it is metabolically very versatile and that it will respond exquisitely to its environment. And therefore, we can have 10 different ways of studying Pseudomonas in the lab. Who has the right model and whatnot is the age-long question. And so trying to understand what are the important physiological and phenotypes of Pseudomonas in the CF lung has been a real challenge. And it's only really been through now, a number of studies that have looked at in situ, whether it's transcriptomic or protein or metabolomic, but in situ from CF sputum or from live patients that really are giving us a better idea, both about what's happening in a human infection, but also how different it is from what we study in the lab. So without going into the differences of lab versus in vivo, I'm just going to highlight a few major characteristics that have emerged from these studies that characterize how Pseudomonas behaves on a physiological and phenotypic manner in the CF lung, as best as we know today. So we know that in a CF lung environment, there are some features that I think most of the studies converge on, is that there are gradients of oxygen at the microscopic scale that will affect, importantly, the behavior, the physiology of Pseudomonas and its susceptibility to antimicrobials. There are limitations in key micronutrients. The carbon sources are altered. There's a large exposure to reactive oxygen and nitrogen species. That there's, of course, antibiotic exposure and selection, and that Pseudomonas can grow as aggregates, biofilm aggregates within the CF mucus. And the responses of Pseudomonas to this environment is that there is certainly an important preponderance of microaerophilic or anaerobic metabolism, and leading to denitrification, and I'll go into that in a few slides, that there's limitation in iron in many cases, that the growth rate of bacteria is reduced with reduced energy production, that central carbon metabolism is altered, that there's scavenging of ROS and RNSs, that there's increased expression of many efflux pumps, and that there is increased expression of exopolysaccharides in the context of biofilm aggregate growth. So what about this physiology and phenotype during CF can be exploited towards new therapies? So the first physiological trait that I want to explore is that this preponderance of the microaerophilic and actually anaerobic metabolism. And so it's been now actually 2 decades or more that people have demonstrated that there are anaerobic niches that Pseudomonas sees within the mucus of the CF, whether those that anaerobic environment is due to oxygen depletion by the host or the bacterial consumption. But the bottom line is that Pseudomonas will undergo anaerobic metabolism. And in order, many of you will know that Pseudomonas is a facultative anaerobe, so it can switch back and forth between high, low, and no oxygen concentrations. And under anaerobic growth, it needs it can denitrify, meaning that it can use nitrate as a electron acceptor in order to generate energy. And so people have been thinking, well, how can we exploit this anaerobic metabolism in order to kill Pseudomonas? In particular, because it's demonstrated, at least in the lab, that our conventional antimicrobials, antibiotics, don't work as effectively in anaerobic bacteria, in anaerobic Pseudomonas, I should say. And so I'm just giving you a brief, very simplified cartoon here of the denitrification. And in here, there are two examples that I'm going to give you of how this can be exploited. So the first one is the first step of denitrification, which is the conversion of nitrate into nitrite by this enzyme NAR. And we know that this enzyme is highly active in CF sputum. And the Diane Newman at Caltech had been working on a compound called chlorate, because chlorate looks like nitrate, and it can be converted by NAR, but now into a toxic metabolite called chlorite. And this will target specifically anoxic bacteria, and it can kill subpopulations that are tolerant to other antibiotics, in particular, for example, tobramycin. And this chlorate is well tolerated in vivo. And to date, where they have been able to push this is that in mouse models, and they've been testing this in wounds, chronic wound infections, it can improve the wound healing and reduce the bacterial burden. But unfortunately, there's also some antagonism that may be happening with tobramycin, as when a combination of chlorate and with tobramycin were tested, these were not synergistic, and in fact, the tobramycin activity might be a little bit reduced. And so at this point, they are thinking of moving this into a human trial, but for chronic wounds, and then whether or not this could be applied to CF remains to be seen. Along the same idea, the next step in the pathway is the conversion of nitrite to nitric oxide. And so again, a number of years ago, people, particularly Dan Hassett, had proposed, well, what about using nitrite as a way to generate a lot of endogenous NO, and this would be able to kill Pseudomonas. In vitro work, they had shown that nitrite killed anaerobic Pseudomonas, as well as in biofilm condition, as well as in mouse infection models. And one of the intriguing aspect of that is that mucoid Pseudomonas was exquisitely sensitive to nitrite, and that there was also a track record in clinical trials of using inhaled nitrite for other, cardiovascular diseases, but maybe, this could be used in CF. So there is actually a phase 1/2 trial in CF that is planned to be initiated. I don't know if it has started yet or not, but it is in the works. And there's also combinations of nitrite and a chelator, EDTA, that is under development for wound infection. Again, this is there are some weak links to this. Similarly to the chlorate, there is a antagonism between sodium nitrite here and aminoglycoside killing. In fact, perhaps a little bit more so than with chlorate, where one sees nitrite inhibit the activity of tobramycin. So I think that is one of the concern about how clinically applicable this would be. Now, if we move on to a different metabolic pathways that have also been, studied for a while, is iron limitation. Now, this has been a target of a pathway of interest, for many, many years, and it has been, quite challenging. The idea behind iron is that we know that it's essential for bacterial growth and many key cellular processes, and that iron chelation has potent anti-pseudomonal activity, both in vitro and biofilms, and as a potentiator of other antibiotics. And the ways to approach it in the most recently have been two ways, either to use chelators or to use a mimicker of iron gallium, which Peter had mentioned earlier on. And so in terms of the chelator right now, there is a phase 1 trial that has been completed for inhaled lactoferrin and a compound called hypothyrosinate. And after many, many years, it has closed, but unfortunately, I don't know that the results haven't been disclosed, and it's not clear what the R&D outcome of that compound is. Now, moving on to gallium, that has also been of great interest, and gallium is a mimicker of iron, but it cannot go through the same reduction that iron does, and therefore it disrupts a number of iron-dependent processes. And the two most commonly studied compounds to date are gallium nitrate and gallium protoporphyrin. And in vitro and in mouse work, it's been shown by a number of groups that gallium, and particularly Pradeep Singh, that gallium nitrate has excellent activity in vitro against Pseudomonas biofilms, as well as in a CF sputum and in mouse study. And this was predictive in a mouse study. And so this has led to a recently, well, actually not so recently completed phase 2, although the results have not been published yet, which of using IV gallium to look for improvement in lung function. And unfortunately, they did not meet the clinical endpoint. And so at the time, which was a significant response by FEV1, but that there were some, in subgroups, a trend towards a decrease in the bacterial load. And so this is pivoting towards looking at inhaled gallium formulations. And as far as I understand, there's a phase 2 that's currently underway for CF. And then more recently, there's been some preclinical work looking at combinations of gallium. And this is actually kind of quite interesting, where they show that the combination of gallium are complementary, that they do not, that they probably target slightly different iron-dependent processes and have a synergistic effect. And in the mouse model, in using intranasal delivery, this showed a pretty striking protection in an acute infection model. So maybe that's also something to be looking at in terms of a combination gallium for inhaled therapy in the near future. And then moving on, in terms of energy production and central carbon metabolism. And so people have certainly described that in various antibiotic tolerant state, Pseudomonas has a very altered central carbon metabolism. And that's has been a group at MIT, Jim Collins, has tried to exploit this. The idea of manipulating the central carbon metabolism by feeding a variety of different TCA intermediates or other carbon sources to see if you could accelerate, among other things, the energy production, and by the process enhancing tobramycin uptake and potentiating the killing by tobramycin. And so there were several years ago, there was a study that reported a fumarate in combination of tobramycin was particularly potent at enhancing the killing of tobramycin, both in biofilm form and this was actually in commercial development, but until I think just around the time of COVID, and then what has happened to the company since then, I think it has been bought up. And so that product is in limbo, but it was actually kind of an interesting thought about formulating tobramycin with a simple carbon source like fumarate. So finally, the last thing that I'm going to mention is the idea of EPS production. And how am I doing for time?
You're fine.
Okay. All right. So Pseudomonas produces a number of exopolysaccharide and work with the collaborators at Cystic and Val Waters, we've been interested in PSL. So we know that eradication of Pseudomonas is challenging, and usually even in the context of using antibiotic therapy, there's about up to 20 to 40% rate of failure. And Val Waters had looked at the isolates of patients that had failed eradication therapy, and as well as some isolates from the EPIC trial, and found that there was a higher binding of PSL, suggesting that PSL was expressed at a higher amount, either particularly when it was grown in biofilm. And then following up in a study with CF sputum, looking at PSL directly, there was also a finding that PSL expression was particularly high during chronic infection, and that in patients that had failed eradication, that there was also a high expression of PSL. Now, PSL is important for the matrix of Pseudomonas, for the attachment, but is also important for host-pathogen interaction. In particular, Dan Wozniak and others had looked at how it interfered with neutrophil phagocytic killing and complement binding. And this led us to look at these isolates and how they behave towards neutrophil antibacterial functions. So we had taken these isolates from the Cystic cohort that Val Waters had characterized. We had screened all of the isolates pre-tobramycin, and those that were persistent versus eradicated went through an assay with neutrophils. And what we had found was that the isolates that came from the infections that were persistent were typically resistant to phagocytosis as well as intracellular bacterial killing by Pseudomonas. And so to follow up on the mechanism and see if we could target this therapeutically, we had chosen two of each group representative isolates, in particular based on their PSL production. And so the persistent isolates were high PSL producers, whereas the eradicated were not. And these persistent isolates here were resistant to phagocytosis, killing. They couldn't activate neutrophils. And in a mouse infection model, what was remarkable is that they persisted. So in the we were comparing here over 4 days. This is not in agar bead, nothing, just in a wild-type mouse. Persistent isolates would persist up to 7, and we did this later on, to 7 to 10 days, whereas all the eradicated isolates were completely eradicated. This was not because these isolates could not recruit or neutrophils or stimulate a proper cytokine response. And so the hypothesis was that the interaction with the neutrophils, particularly the opsophagocytic killing, was impaired. And this is where we became interested in looking at some of immune therapy. So this is a bispecific monoclonal antibody that is under clinical development by AstraZeneca. And in preclinical work, they had found that it had improved bacterial clearance and survival in acute pneumonia model, among other things, and that they had completed a phase 2 trial for ventilator-associated pneumonia, which unfortunately did not show a superiority, but it was in a ICU high mortality type of patient population, and right now they're doing a phase 2 on non-CF bronchiectasis. So we wanted to know, could this be used in CF, particularly in these isolates that were persistent and were high PSL expressors? And so we took these isolates, and this is an in vitro assay, and we did show that all of these isolates could be potentiated with the antibody, that there was, on average, up to a 4-fold increase in phagocytosis and an about 2.3 or so increase in antibacterial killing in vitro. And then we're proceeding this to look at this in a mouse study. And this is starting out with simply prophylactic monoclonal antibody. And this is the antibody tested against 2 of the persister strain. And what you can see is that it is superior to IgG control early on in terms of the bacterial clearance. But unfortunately, for the time being, at later time points, although there is a reduction, there is we do not see complete clearance. And we do not see a statistically significant better clearance than the IgG control. So this leaves us, does it have a role in combination with antibiotics, which is what probably you would expect to use, and then whether it has a role once the infection is established also remains to be determined. So that's work in progress. So with this, I'm just going to leave you with a few concluding thoughts, which is that I think that there's a tremendous body of work that has been looking at various physiology and phenotypes that contribute to the infection persistence, and it's very highly complex. And although many approaches have the potential to overcome the barriers that we associate with ineffective antibacterial treatments and with promising preclinical data, it's been incredibly difficult to translate into clinical findings. And this is due to a variety of both biological reasons in terms of the pathogenesis and the complex biology of Pseudomonas, financial reasons because of the lack of incentives and companies folding and going bankrupt, and then clinical hurdles, which now have certainly increased in the era of highly effective treatment. So with that, I just leave you, from here, where do we go? So my first question, certainly as a clinician scientist, is thinking kind of what are the clinical unmet need for treatment of Pseudomonas in the era of triple therapy? Do we need more systemic treatment for pulmonary exacerbations with multi-drug resistant or carbapenem resistant Pseudomonas? Probably less so than before, as there are fewer and fewer exacerbations. Do we need more maintenance therapy for chronic Pseudomonas? Again, perhaps less so than before because, most patients, although they're not eradicating Pseudomonas, their bacterial load is significantly lower. And patients are not keen on taking maintenance therapy. So perhaps is it leaving that the main unmet need is better eradication therapy? And if so, how are we going to take all of this knowledge in microbiology to come up with a new therapy? And more importantly, what are the models and what are the clinical endpoints that will allow anybody or any company to come up with an effective therapy? I don't have any answers, but just some food for thought. And so this is a work I'd like to thank the students in my lab who have done the work. The antibody was provided by AstraZeneca and Val Waters for being a collaborator. Thank you for your attention.
Okay. Questions for Dow, and if you have questions for other speakers, feel free. And I've got a few questions for, I think, other speakers as well. So.
Hi, Dow. Great talk. I was just curious, have you looked at those persistent Pseudomonas to see in their genomes, are there any mutations that might explain why they're making more PSL?
So I think that that's a question probably for Val. I know that they have done whole genome sequence analysis of all of these persister strains. Whether they had identified mutations specifically to explain the PSL overproduction, I do not believe. Perhaps in one or a few isolates, but not across the board. No.
Cool. And a related question, do you know for those persistent isolates, were those the first bugs isolated from those patients, or were they like, a year or 2 years down the road?
So the definition that was used, so this was piggybacked on the clinical sort of observational cohort study that sick kids have been running. The definition that they have for new onset in Pseudomonas is that the cultures have been negative for the preceding 12 months. So it is possible that in some of them, at some time in the past, they did have Pseudomonas. And in certainly, when you look at the phenotype of these isolates, occasionally you do wonder, they have more chronic type phenotype, so did they actually have the chance to evolve already, even though this was the first isolates that we had?
Yeah, cool. Thanks.
Hi, enjoyed that. Thinking about the NAR gene expression levels, high levels of NAR, high levels of nitrate, was there a consensus on the source of that nitrate in the airways? Where's it coming from?
Where it's coming from?
Pseudomonas is seeing lots and lots of nitrate.
Yes.
Where is it coming from?
I don't know.
Does anybody know?
No. I don't know.
I heard neutrophils. Yeah, neutrophils. Thank you. Yeah. I think I'll just make a comment. I think at the last part of your talk about sort of where do we go and what do we need, I think just two comments about the multi-drug resistant. I think one is, we've been talking at this conference, there are people that are not modulator eligible, so they're going to continue to have, progression of Pseudomonas, presumably. So it becomes a rarer group, but still really important. And I think the interest in the phage kind of answers the question, too, about how many people are interested in treatment of multi-drug resistant, because, people still have it. So, yeah, it's great. Other questions? I will take this opportunity then. If you want to, you can stay up here. You can stand, but I'm going to ask Peter a question just to jump back to our first. Put you on the spot. But somebody asked, are there any potential side effects of blocking ferroptosis in people? Yeah, you can go there. You can either one. Yeah.
Okay, so potential side effects of blocking ferroptosis. So it is a form of programmed cell death, so it could be important under certain circumstances. We don't know. They are actually pursuing activating ferroptosis as a way to treat cancer. So, that suggests that ferroptosis can be important at times. So that's a thing that we'll have to consider going forward.
Interesting. Yeah. Thank you. And then this is a question for Daria. As a CF patient, is there a recommended isolate collection for providers in preparation for possible future use?
Yeah, I want to highlight, again, like John LaPuma's fabulous collection out of Michigan, following all these Burkholderia patients over time. I wish that existed for every pathogen. At our own center, we do a ton of banking of isolates from patients who are frequent flyers and come, and working with our CF physicians to try and, every time they see a patient and they collect a sputum and send it for culture to let us know so that we can run to the micro lab and grab it. But in terms of public repositories, John's is the only one that I'm aware of, but I wish that they existed for other pathogens as well.
Okay. I think that's all. Any last comments? This is great. We had 4 outstanding speakers, so it was excellent. Thank you guys so much.